So here we have aerobic cell respiration, and it's called aerobic because of our oxygen here. So aerobic, without it we have anaerobic respiration or fermentation, but we're just going to focus on the aerobic equation today, and this is from start to finish. So this is the glucose here that we begin with, and this has to undergo glycolysis first in the cytoplasm, and then once it's broken down into roughly half, 2-3 carbon molecules called pyruvate, it enters into the mitochondrion, undergoes a link reaction in the Krebs cycle, and then all these banked up electrons are used to generate lots of ATP, our energy-rich molecule. So we could actually put that on here too. The whole point of cell respiration, if you like, I kind of just do a big arrow here, is to go from lots of adenosine diphosphate, so it's got 2 phosphates and an inorganic phosphate, and to join them together to make adenosine triphosphate. So that's our 3-phosphate molecule, ATP, and that's our, kapow, that's our energy-rich molecule that's used by cells to do everything. So DNA replication, movement, all sorts of things. We need ATP to power our cells, but in order to do that, it's like our little battery that we recharge. We recharge it up by breaking down organic molecules like glucose to build ATP, and then ATP gets used in cellular processes and converted back into this low energy form. So we need to constantly keep rebuilding up ATP. So aerobic cell respiration takes that glucose and it combines it with oxygen to make, let's put carbon dioxide over here, and of course water. All right, so if we wanted to look at this, using this equation, we wanted to do some measurements on cell respiration in the laboratory, the sorts of things we could look at glucose being consumed. It's a little bit trickier. You can use their kind of little dipstick tests, or if you had a blood glucose monitor, you can sample some solutions and work out how much glucose had been used up by some respiring organisms. You could look at the oxygen being consumed because they're using up oxygen. So if you have a sealed environment, you could look at how much oxygen is being taken in. Or again, if it's a sealed environment, we could look at the carbon dioxide coming out as a result of that. But don't ever try to measure water. That's not a very good measure. Even though it comes directly from this equation and it's a product of cell respiration, animals and plants have, or any other organism has so many other ways that they lose water. Think about a leaf, for example, and it loses water from transpiration. So the water is evaporating out of that leaf anyway from other processes. So you're going to get really erroneous results if you try to measure water, assuming it's only coming from cell respiration. So ignore that one. So possibly glucose, but your best two for most school laboratories is going to be oxygen and carbon dioxide. The other thing that is possible, but it's indirect, is you do also get heat that's given off. And because most of this energy, we try this sort of process of breaking this glucose down is through lots of controlled steps that are coupled to other chemical reactions to charge up this ATP. But inevitably, some of that energy transformed is actually going to be as heat that then dissipates out. But it is more controlled. I mean, the chemical equation here is actually the same as combustion, as burning something. But when you burn something, all the energy pretty much is becoming heat, and it's not very controlled. So respiration is a kind of a controlled combustion, if you like, in order to harness lots of ATP. But you do get some heat. So you can indirectly measure the rate of respiration by looking at heat given off by something, maybe putting something in a vacuum flask, a thermos, popping a thermometer in and looking at the heat change. but it's a little bit trickier to control as well, because if you're going to put your thermometer in, or things perhaps are producing gases, particularly anaerobic things that have an unequal number of gases in the equation, then you can have some trouble sealing your container. So it's okay to do heat, but if you can do one of these gases, that's much better. So in order to do that, you can put samples in a jar like this. If you've got the equipment at school, this is a sealed container here, and you can put in some of these probes. This one here, this one is an oxygen sensor, and you know, the lights will change. This one tells you it's wireless, like it's red when it's ready to connect. It'll blink green when it's connected and yellow when it's recording, and there's a battery indicator light as well, kind of a traffic light system. And that can connect in and has a perfect seal to a container like this. So you could have a sample of something respiring at the bottom, and then you can quite quickly get some good data that's quite precise to measure the oxygen, the change, because there will be a change in composition of the gas in here as oxygen gets used and carbon dioxide gets made. So that's a possibility. And the sorts of things you could pop in here, things that are respiring, in theory, pretty much anything living will respire. But ethically speaking, we want to be careful about animals. Classically, there have been experiments with invertebrates. We tend to sort of be a little looser with our ethical kind of guidelines with those, but just think of any animal, doesn't matter what it is, try not to stress it, and you want to make sure that it doesn't actually get so depleted with oxygen that it's in any risk. So a good thing that you can use though is germinating seeds. So a seed doesn't do photosynthesis, it hasn't grown any leaves yet, but it has got a store of starch inside it that it's breaking into sugars and then respiring really rapidly so it can grow new shoots. So things like mung beans and things like that are good seeds. You just wet them and keep them warm, make sure they're well oxygenated, and they'll start to respire. So you can pop those sorts of things in there, and then maybe investigate putting this under different temperature conditions, or pre-treating the seeds, perhaps to affect change the outcome there. The other thing, you can also use a carbon dioxide sensor. So in the same way, you can pop that into a sealed flask, and just like the oxygen one, so this is a carbon dioxide sensor, you will then see the change in proportion of carbon dioxide in this air sample that's trapped in here. So good for getting quite quick data, quite precise and pretty easy to do replicates. Just be careful when you're setting this up or refreshing things, or you may want to take your container outside to get an atmospheric level, oxygen and carbon dioxide level before you begin. Now the other one, and one that sort of comes up in exam questions a lot, is this one here. So this one is called a respirometer. And I'm going to take you through this. Let me move this piece of paper and show you. Right, so here's our respirometer, or at least part of it. I haven't actually attached the tubes yet. But this section here, this is called our U-tube manometer. And you'd put some liquid in here. Usually you'd pop a bit of food colouring in to make it a bit easier to see, or sometimes a bit of detergent to change the surface tension, just a drop or two in, make a mixture first. And you can take these bits off to be able to introduce some fluid in here, and you might use a syringe like this one to be able to put some fluid in. And so it doesn't really matter what amount you have, you just want to have sort of a kind of like a little bubble or a little zone with some fluid in here, just visually, so you can track any changes. What you're going to be looking for with this setup is you're going to be connecting some test tubes or boiling tubes through the back here with a sample of something that's respiring. Now the principle upon which a respirometer works, and this one kind of sinks to the bottom a bit quickly, but what you're going to do in this one, in your test tube, you're going to put a sample of the organism that's respiring. So say for example your mung beans again, a bit like we were showing with this container. So you're going to be putting a living sample in here, and then the difference with this one is that you're not going to have both the, you're going to have the carbon dioxide given off in your air sample. You actually want to get rid of it. You want to absorb it. Well, I might have moved that too quickly. If I bring that one back, remember, normally if you've got oxygen coming in and being used, you're also getting carbon dioxide given off, and you've got the same amount. You're going to have 6 moles of each of those. So the total volume or amount of gas or air particles, if you like, that are trapped in that, if that's sealed with a stopper, that would stay the same. So in a respirometer, what we want to do is we want to get rid of this carbon dioxide. We want to absorb it so that there is a change in the volume of gas as oxygen gets used. So we want to pull oxygen in and then trap the carbon dioxide down the bottom in a liquid or even a solid. We want to absorb it using something so that we can see a change in the volume of gas. And on this side, in our manometer, if we have some liquid that's in here, if air gets pulled down into our test tube, if we have air being drawn down in here and we're absorbing the carbon dioxide so it isn't replacing the oxygen, we're going to get a change in volume of gas. The air is going to be sort of sucked in here, if you like, which means if there's a little bit of liquid inside this tube, that's going to be pulled along too, and we can actually record the change that occurs. So that's the principle behind a respirometer, and I might draw that as well so we can talk through it a little bit more. But just so you can see the equipment, you do, let me connect. Okay, so here's our respirometer and our YouTube manometer at the front. And then if you have a look, so it's backwards for me, you've got your test tubes at the back. Unfortunately, that one's got a little chip out of it. But you've got 2 of them, and then they're connected through here, and you'll have some liquid in here. And really what you're doing, you're actually comparing the pressure in both sides. That's going to cause your liquid to move. And the reason you've got 2 of these test tubes then is you can actually have one side as a control. So you can pop something to soak up the carbon dioxide in the bottom of both. And you might find that it pulls a little of the carbon dioxide out of the atmospheric air anyway. So to correct for that, you can have one side with no living specimen in it. And just some water equivalent to the volume of whatever you're measuring, and the other side with your sample, say of your mung beans. So you'll have them both in there and then connected, and we can see them kind of fighting, if you like, for the air pressure, and that'll move the liquid around in that one. And putting the test tube on here, you need a rubber stopper, so you've got no other atmospheric air that's able to come in. Now this one has a 3-way sort of tap or valve on it. So what you can do, you can take a piece off the top. So where you've got one of these points on this blue tap, you've also got a little hole in here. So that's going to allow air in and out. So you're going to have, like this, for example, showing that there's a hole to my test tube, so that oxygen can be drawn in through there. And then also at the top that it's open, but at the moment it's closed to this manometer part. So you'd have to move that around that way. So now that's pointing down, show that that hole is open, and this one pointing across, showing this hole is open, so that the air inside this section of the respirometer can be drawn down into the test tube. Now, when you do get ready to start and you put these, connect them both up, you can pop it, if you've got it in a test tube rack, you can then sort of dunk the whole thing in a water bath if you like, because temperature is a good one to investigate, and this system is safe to be submerged in water up to sort of this, well, to the bottom of your stopper. You can submerge it to sort of that height. So your sample is quite well covered. You want to give it a little bit of time to equilibrate as well. You want your organisms to kind of start respiring and reach a steady level and be in the conditions, have it all come to temperature before you start recording. Sometimes that means that whatever liquid you've got in here is it's not equal on both sides. So what you can do then is add a syringe, set it to sort of the halfway point is usually good, but depends what you've got. Make sure, again with this three-way tap, that you've got that pointing up so that there's an air hole connected. Otherwise you can just cap it off when you don't need it, or turn that around. That means those two are open and this one's not. So now all three are open. And then you can use the syringe to push in more air and push the liquid back down if it's too high on this side, or the reverse, pull it up and bring it back around. So in doing that, you can actually sort of reset your respirometer or make sure it's at a good starting point. It should be equal to start with. It also means if you want to take lots of replicates, you can easily reset it by using that syringe up the top there. Drawing's not really my forte. That took me longer than I'd care to admit, and it's still wonky. But here's my sort of diagram to show you this respirometer, although I've just kind of drawn these test tubes to the side so we can see them a bit better. So let's say here, sorry, that should be, I already had that written, but that's that bit. That's the manometer. So you're going to have some sort of liquid that you place inside. So let's say it's sort of here. Just kind of, this is a good thing I've got my kids' set of coloured textures here. Okay, so you'd have some coloured liquid in here, and then we've got our sample. So this might be our mung beans or whatever it is. So this is our respiring organism or organisms. Yeah, and things like seeds are good because they're pretty small, so you can get fairly consistent kind of quantities of them by weighing them out, because that's going to be a controlled variable. So we've explained some of these things already. This is our respirometer, this is our test tube, our sample. Now we mentioned we needed to absorb the carbon dioxide, because remember we want the air to be drawn in by using up oxygen, but if we were giving off carbon dioxide, that would just push it back out again, because the oxygen used is the same amount as the carbon dioxide given off. So we would see no change to the total volume of air in there if we didn't get rid of the carbon dioxide. Thankfully, carbon dioxide and water makes carbonic acid. So if we put a strong base in here, that's going to absorb all of our carbon dioxide, most of it anyway, we hope. Let's just draw a line down here. I can maybe colour that in. So this down the bottom is going to be either a solution of base, or it can be like solid pieces, but most typically sort of in these tubes that we might use. And see too how like this part is actually made of mesh so that the air can circulate, but you don't want your organisms into your quite sort of dangerous, toxic, strong base down the bottom, or you'll kill them. You won't get any respiration happening. So this down the bottom here, because I've got more room on this side, I'll label it over here. So this is going to be a strong base which absorbs our carbon dioxide, and that's on both sides. Now, the strong base that you could use We often use potassium hydroxide. So you might see it come across that. Or you might use sodium. Or you might see, if you see a combination of that or a solid with some lime, we call that soda lime. So let me just add that in case you see that. So you might see labelled as one of these solutions, or soda lime or caustic soda, or a few different names, but it has to be a strong base because it's an acid-base reaction with the carbon dioxide. The other thing you can do to make this even more effective, if you take a piece of filter paper and kind of roll it up, I'm not sure how I'm going to draw this very well, but if you just get kind of a little, I don't know, curled up kind of bit of paper, that'll do, won't it? I told you, art not my thing, but I do like colours. Okay, so if you have a little piece of filter paper in there, it just kind of helps sort of soak up capillary action with the fluid you've got in there. It gives you a bit more surface area and just makes the absorption of the carbon dioxide a bit more efficient, because you want all the carbon dioxide to be absorbed so that that air does get drawn in. All right, now why we've got the two we mentioned before. There is carbon dioxide in air anyway, so if you just put like some respirometers that are more simple, they just have one side. So if you might just have a straight tube, and you can do that. You can just connect, and you don't need the syringe. The syringe just helps you reset it. You can just have a plain stopper and a single tube, maybe a piece of capillary tubing, and then you can see a dot moving in, and you can measure that. That's fine, that works. But if you want to account for the fact that there's carbon dioxide already in the air, because if you had something dead in there that wasn't respiring, you would still see the little water droplet move down and air being drawn in, because the carbon dioxide that's already existing would be being absorbed. So to counter that, by having this control chamber over the other side, or sorry, I've just realized my soda lime stopped off the bottom because of where my camera's positioned. So if you have an empty chamber over here, it still has a sample of atmospheric air. So both of them are going to have carbon dioxide drawn out of them. And then if they were both the same, this water wouldn't move. So even though the pressure's dropped, it's relative to each other. It's a comparison. So it's only when the pressure will drop a bit in both as atmospheric carbon dioxide gets absorbed, and then if something's respiring here, it will continue to pull oxygen in so that the pressure will drop even more, and we would see this liquid going this way. So we would see, sorry, I should have brought some white out, but that would be going down on this side. Maybe I'll just put an arrow down that way and up this way. So the bubble moves in towards whatever is respiring. So the sorts of things in an exam question, you might be asked something about this setup, and there are lots of different variations on how a respirometer can be done. But key things are having something to absorb the carbon dioxide, so it's going to be a strong base. So some of these options here. This, I'm just showing this as being clamped off, by the way, but the chambers have to be sealed so that you get a change in air pressure. But a feature here like this syringe is to reset or establish, like get our liquid in the middle before we start. What else? They're probably the key things. And just referring, I suppose, back to that equation, the air gets drawn in and the carbon dioxide that comes out has to be absorbed by something, so that we can see the change in oxygen or the air being drawn in. I'm saying air in quite lay terms there, but it's just the oxygen from the air. But the whole volume of gas particles is going to decrease as we draw in oxygen going down here and being used by these respiring organisms, and the carbon dioxide coming out is going into here, and it's getting dissolved in this liquid and reacted with the base so that it's no longer part of the gas particles in here. Okay, hopefully that makes sense and that's helped explain respirometers a bit.